Good morning, everybody. Welcome to our Coulter seminar series that we have every week. And today we start on the technical leg. And it is my great pleasure to introduce to you Dr. Cynthia Chestek. I think I met Cindy at one of the DARPA presentations where she spoke very enthusiastically with a lot of passion, as she always does. Dr. Chestek was highlighted last year by the Society for Neuroscience, which reaches, I don't know, 100,000 people or so, as one of the entrepreneurial women combining neuroscience, engineering, and tech. And indeed, that's what she has been doing since she was an undergrad. Those undergrads who are online, you may ask her, what was she doing with aplysia during her undergraduate? And apparently that's what got her into research. She got her bachelor's and master's degrees in electrical engineering from Case Western. I got my doctoral there, so Case Western Raha. And then she got her PhD at Stanford. Very interestingly, the project that she then did was, again, as a postdoc, was this brain gate clinical trial where these electrodes, very fine electrodes, were put in actually in human subjects in the brain who had ALS or the Lou Gehrig's disease. But maybe she will get an opportunity to tell you more about it. But without taking any more time, I am looking forward and to listen to her presentation that will follow. She is interested in electrodes, she is interested in neuroscience, she is interested in movement, she's interested in making a difference in people who have lost function or have neurological disorders. She really is a true neural engineer, bringing all of these things back together. So welcome, Cindy, it's all yours.
All right, thank you very much. So I'm gonna go ahead and leave my video off for now, just because we're having a little bit of internet difficulties, but hopefully we'll just charge ahead, and I apologize if there's any further bandwidth issues. All right, so today I'm gonna talk about the neuroprosthetic control of finger movements. So my lab focuses on motor neuroprosthetics, and what that means is that we use signals that we actually get from all over the nervous system to control external end effectors. So, and I think my lab is sort of different from others in this regard, is we have the huge privilege of working with a lot of clinical collaborators at the University of Michigan. So we get to use brain, nerves, and muscles to try to control motor devices. So for the case of trying to get signals from nerves or muscles, and there we're trying to think about how to develop better controllers for prosthetic hands. When it comes to the brain, we're really thinking in terms of how do we get a control signal for somebody with a spinal cord injury. So the goal there would be to eventually actuate a paralyzed limb. So I'm gonna, my lab sort of spans a range from, you know, things that are, you know, in people today, sort of all the way back to stuff that it's gonna be a while before we sort of see it in a clinical setting. So I'm gonna order my talk here from the thing that is the closest to being out in the world, and then I'm gonna go back through the things that are a little bit farther away, and, you know, at the end talk about carbon fiber electrodes, which I think are, you know, hopefully the future of these other things. All right, but first I'm going to talk about nerve-controlled prosthetic fingers. So The Empire Strikes Back came out in 1980, which is 40 years ago. And so the idea of just taking a prosthetic hand and hooking it up to the residual nerves is very old. You know, and there were lots of very smart people working on this for a very long time. So why don't we have nerve-controlled prosthetic hands? And I think the answer is that no matter how you look at it, the physics is really not in your favor. You're talking about trying to get a signal from, you know, an extremely small, extremely well-insulated wire. So, you know, if you try to put a cuff around it, you don't have enough specificity. And, it's just, there's certainly people recording from nerves, including at this institution, but it's really challenging to get these really big, multi-year lasting signals that you're going to be able to use in a wide range of contexts. So in fact, it's, in our group, it's so hard to record from a nerve. Our surgeons found a way to take a nerve and turn it back into a muscle. So we call this surgery the regenerative peripheral nerve interface. If you're familiar with TMR, it sort of shares the principle of, reinnervation. But how this works is, you divide down a severed nerve from an amputation into individual branches, and our surgeons sew small pieces of muscle around the end of those nerves. And then the nerve reinnervates into that small piece of muscle. And, what was a, single digit microvolt signal in some cases now becomes, hundreds of microvolts. So we're using the muscle as a bioamplifier. So I'm going to show you one. So we did this, this was developed over in plastic surgery at Michigan and tested in rats. In my lab, we did non-human primate work, just amplifying nerve signals. Everything went extremely well. And so we went pretty quickly to humans. So I'm going to show one surgery picture on the next slide. And our human work was just published earlier this year. But so this is what it looks like. So there's, little bits of muscle attached to the end of nerves, and you can divide down the nerves to get more specific signals. So, that looks fairly dramatic, but those are about a centimeter in diameter. So it's still pretty small. I'm showing you here a person with a transhumeral, or the nerves are transected at the transhumeral level. So most of the finger muscles, essentially all of the finger muscles are gone at this point. So, you wouldn't have access to them for controlling a prosthetic hand. And the easiest way to see what the surgery does, I think, is under ultrasound. And I should say this person in particular, he had his amputation for over 15 years. And he said he felt like his hand was frozen in a block of ice, that he just didn't feel like he could move his hand. And so you can see here, if we look under ultrasound, and afterwards he felt like he could move his fingers. And so specifically here, we asked him to move his thumb. And you can see there's now a wiggling thumb muscle inside of his tricep area. So we created a thumb muscle. Similarly, on the ulnar nerve, we got the biggest flexion for small finger movement. And then we also asked for anatomically unexpected movement. So on the median nerve, we don't expect small finger flexion, but you can see the other RPNI flexing from off screen. Similarly for ulnar does thumb and small finger. So we asked for middle finger flexion, and we didn't get it on that muscle. So I guess I have one more surgery picture in here. We then moved, we went to the FDA and said we want to do this in a sort of semichronic preparation, start with wires going through the skin before there's an implantable device. And, we obtained approvals for that. And we have two wonderful, amazing volunteers that volunteered to have this done. And they have, they had already had this RPNI surgery. And they have a connector on their arm that, we can go into the lab, they come in, we can hook them up, and they can start controlling prosthetic hands. So first, let me start with the RPNI signals, just so you can sort of see that these are big, healthy signals. So from our first participant, what you're seeing here is EMG traces for these 4 different movements, and the top and the bottom are RPNIs, or the 2 RPNIs that this person had that were on motor nerves. So on the ulnar nerve, you can see nice, big, healthy signals for thumb, ring, and small finger, which makes sense for the ulnar nerve. The median RPNI just loves the thumb, so you can see the specificity of the signal here. There's virtually nothing for index, ring, and little fingers. So the signal to noise ratios, it's 33 and 149. And I should say that these signals have spoiled us for all other kinds of signals. You know, even like brain signals are pretty good, but like you never see this kind of signal to noise ratio. And so, you know, I'd like to think our machine learning is pretty good. I think it is, but like these signals are just really, really big, and that makes all the difference. Okay, so what does this look like? So in practice, for every person that comes in, there's some signals that we're getting from the nerve, and there's some signals that we're getting from the residual muscles. So if we're going to implant somebody, we implant them in the best possible way for them. And then, so, you know, since we published the RPNI paper, now we're just using all of the signals together, and we decode prosthetic movements in a couple of different ways. So the first way is classifiers. Right, and so what you're seeing here is we're attempting in 2 participants to classify 10 different movements. And so this is a confusion matrix where 100% correct would be all yellow down the diagonal. You can see for 10 movements, offline we're at 99 and 97% correct. Online is always more challenging. It's 92 and 86% correct. We're doing this with what's called a hidden Markov model, which lets us be very responsive and you know, switch very quickly between these movements. And so let me play a video of what this looks like. And I'm playing this in animation because the hand can't actually go this fast. So we are controlling the hand in front here, and she's controlling the hand in back. And this is the 10 different movements. They'll just play a few. So not perfect, but it's 10 movements and it's pretty darn fast. Okay, so this is you know, this is more challenging to do with a physical prosthesis where somebody is actually moving around. So we did a lot of experiments to sort of, you know, figure out what still works, you know, nearly 100% when we're doing that. And also the hand can't actually make 10 movements. But this is what it looks like with, you know, the 3 grasps that the DECA hand can make in multiple different postures. He's going to do a fist, a pinch, and a point. And so he can, and by the way, this is calibrated, not trained. So, you know, we take a 5 to 15 minutes worth of training data. He made 1 mistake there at the end. And then they can just go online and start using these. Okay, let me start one video and I'm going to talk over it a little bit because it's long. This is our second participant, and she's going to move all 5 fingers. So you can see here, you know, it's going a lot slower than that animation. So she's going to do all 5 fingers correctly sitting, correctly standing, and then she'll make 1 mistake when her hands are at shoulder level. You can see this is going a lot slower, and that's because the DECA limb it's very cool. It's a marvel of modern engineering, but it's very heavy. And so our second participant couldn't lift it. Our first participant could barely lift it for long time periods. And so we're really grateful for OSER for helping us out with the hand, but this is the small-sized OSER hand. So now this is a very realistic clinical hand. And that's how fast it goes. So, in practice, we end up using a lot of classifiers with our participants because, you know, the hand doesn't move fast enough for continuous control. So we still do continuous control, but we do it in animation. And this is actually, you can see on the date, this was, you know, the first week after surgery. I had just, this is just a single degree of freedom to start, but I had just told our participant, okay, you know, we might have things to debug. You have to bear with us if it doesn't work on the very first try. And he just started moving the thumb and said, hey, it looks like it's working. So, you know, it's a calibration, not a training. So this next video I'm going to play is now 2 degree of freedom thumb movement. And I'll say that this is something that you really need the nerves for. This is something I don't know how to do if you didn't have access to the nerve signal. So my student, Phil, is, who's from Florida, by the way, is holding the target here, and our participant is moving the thumb to the target in a 2 degree of freedom workspace.
Down, up, over.
And so let me just leave you in this section with basically, where we're at right now with continuous control. We have 3 degree of freedom control that's pretty good for 2 degree of freedom thumb and index finger.
Actually, almost choose where it's touching the thumb at.
So watch his thumb here. He's going to move it out of the way, use his index finger to pull a block forward, and then move the thumb around the block. And I think that, our participants actually do prefer, and this is continuous control within a grasp, which is, what the Oster hand can do. So you'll see here that, at some point at the third one, she makes a mistake, but she doesn't, because it's not a classifier, she doesn't make the whole mistake. She can stop the hand halfway and open it again. And I'll say our participants do prefer the continuous control, even though the grasp classifier with 10 grasps looks really cool. But it's really better to be able to sort of reset, orient your thumb, and things like that. All right, so now I'm going to switch gears to the brain. And I like starting, so here we're no longer thinking about amputation, really. We're thinking about spinal cord injury, where there's no meaningful signals in the nerves, and so we have to go to the brain if we want to get a viable control signal for stimulating a paralyzed limb. So, still we do this with the Utah array, which is, it's a bed of needles that goes into motor cortex. It looks kind of intimidating there, but it's about, 1 millimeter deep. So it's actually a very small device. Here's what it looks like on the surface of the brain. And I like to start by playing a video this is from 2016, but things were actually working this well starting around 2012. And so I just want to play it because I'm not sure everybody's seen it, but this is a monkey typing Shakespeare, right? So, and this is from the lab I did my PhD in at Stanford. And you can see, so the monkey's not seeing the letters. He's seeing the white ball and the green target. So he's just moving the white ball into the green target. But the letters are for us, and he's typing Shakespeare, and he's getting to about 13 words per minute. And this is entirely monkey telepathy. So these systems are actually working pretty well. And I think that, so this is making very good progress transferring to humans, over the past decade. And so I think when I started my lab, I was trying to think, okay, well, so what's not quite ready for humans? What's something that we can't really do yet in humans? And we decided to try to focus on getting to multi-degree of freedom finger movements. All right, so what is the state of the art in humans today? So I think it's this amazing study from Baloo Ajiboye at Case Western, where this is a person with a cervical level spinal cord injury. You can see he has 2 Utah array connectors on his head, and he also has wires percutaneously in his arm to stimulate the muscles. And he was able to use this system to do things like lift and grasp a fork, and lift and drink from a coffee cup. And a few degrees, so while we obviously want to get to more degrees of freedom of control, this is already something that we don't know how to do really any other way. And this would be something that I think a certain number of people would be very interested in taking home, even if it requires a brain surgery. So, the problem with this setup is that there are 232 wires going percutaneously through the skin in order to make this happen. And on the outside is a computer cart, right? And that describes 99% of BMI experiments that have ever been done. And so, the first question I always get is, why don't you make it wireless? And so people have tried, but these numbers are daunting, right? So 50 megabits per second and, 232 wires is far beyond the capability of any implantable device that's ever been made, right? An advanced pacemaker is going to have 32 channels. Right. So, even if you had the feed through, the power consumption is nearly impossible. And if you say that you're going to do it the same way that you do it on the cart, where we process what comes out to 50 megabits per second worth of data, it's impossible. Like you can't win. You can't build an implantable device that can process 50 megabits per second on a reasonable battery inside the body. So in our group, we're proposing a new architecture. We're not the first to propose it. I think we're the first to show that this will always be as good or better at as processing spikes. So we'll always be as good as better as what we're doing currently. So let me explain. Again, so I'm going to tell you about reducing the power consumption, but what I'm not going to tell you is that we're giving up the spikes. So we love having spikes. Spikes are very specific to individual finger movements. If I put an electrode in the brain, I'm going to record these bipolar waveforms that come from individual neurons. And, I need that to get the kind of specificity that you saw in those videos. Okay, so what if I told you there was a way to filter this signal that will just give me, that red neuron there and very little of the other two neurons? My student, Sam Nason, just published this in Nature BME, and let me just sort of take you through why what we end up with is a single unit signal. All right, so what do we actually have there? It's basically a 1 millisecond sine wave. Right? So with some frequency components that are a little bit above and a little bit below. So if we take a Fourier transform of those spikes, we end up with something that actually has a large percent of the signal power below 1 kilohertz, right? So if we then filter this signal, which again, we definitely weren't the first to do, we get something that ends up actually being, extremely well correlated with that single unit. Right, so this is actually simulated data that looks a lot like real data, and it's got a single neuron. And you can see here in green is how most BMI systems work, where you sort of you have a threshold for that spike, and in blue is the spiking band power. And so that is filtered between 300 and 1,000 hertz. And you can see that it's actually just following along with the spikes. And if I average that over a time window, I'm going to get something that is correlated, like precisely correlated to the firing rate of that big single unit. Okay, so that's all well and good if there's a single neuron on my electrode, but what if I have a more realistic channel where there's multiple neurons? And even worse, or what if I have a channel that has very low signal to noise ratio? And this is where this technique is really interesting. So when you think about it, I actually am getting power from the up and the down side of this cycle. The whole waveform is contributing power. And what that means is as the signal is smaller in size, the spiking band power can actually pick it up after the threshold has been lost. So here you can see my true firing rate is in green. Threshold crossings have lost the signal. It's too far in the noise. There's a signal to noise ratio around 2. And you can see the spiking band power, like if you follow this blue line, you're actually still picking it up. And in practice, so in this graph on the left, you can see this is sort of, the performance. And for spiking band power, it is penetrating more into the lower left of this graph over here, which is lower signal to noise ratio. And so this explains why, multiple groups have already observed that this signal is suspiciously good for LFP if you just sort of filter in this 300 to 1,000 hertz range. Okay, so let me explain one more thing before I show you our BMI performance, and that's what we're doing on the algorithm side. So there was, I had the privilege of witnessing in, 2012, the development of the Refit Kalman filter. So if you were a monkey in, say 2009, trying to control a cursor with a computer, with a brain-machine interface, so a Utah array and motor cortex, you are stuck with the performance on the right, and that is a straightforward Kalman filter, right? So, okay, so, and then, you know, basically 2 years later, let me just figure out my animations here, we have what's in the middle. And so how did that happen? So basically, it's a retraining of the Kalman filter matrices that's based on the assumption that the monkey can always see where the cursor is and is always trying to get it into the target. And so what I mean by that is we assume that the, you know, so if he's going this direction, we assume that the neural activity actually reflects intention for this direction. And then we retrain our linear regression. So there's no target information that is then included in the algorithm. We just redo essentially one of the linear regressions inside the Kalman filter, and we get a much better fit. And then that goes from what you see on the green to what you see in the middle, which, you know, on a good day approaches native arm control. All right. So in my lab, we extended this to finger movements. So this is from 2018, and this lets me explain our monkey task. You can see the monkey has a little manipulandum here, and there's doors for the fingers or finger groups. Usually we do index finger and middle ring small finger together. We started with a single degree of freedom finger movement. So you can see here, this is brain activity decoded to control that animated monkey hand. If we do our job well, the monkey doesn't realize he's sort of gone into telepathy mode. And now let me show you much more recent data for 2 simultaneous degrees of freedom. And so here we're going to be using the Refit Kalman filter for 2 degrees of freedom at the same time. And this is using that spiking band power technique. So all of this data is sampled at 2 kilosamples per second, much lower power consumption and processing than it would have been otherwise. So first, let me show you on the left, that's his real hand. So that's how well he can do this task. Those fingers can either move 1 at a time or 2 at a time. They can go in the opposite directions or the same direction. These are 2, random degrees of freedom. Here on the right is decoding with the Refit Kalman filter, and again, using that low power technique. And you can say that, and you can see that he's actually can acquire these targets slightly faster on the right. So I just want to specify, we are not saying he can do better than able-bodied. The monkey still has to use this manipulandum, so it's still not quite the same. I guess the manipulandum poses less of an issue in brain control mode. So, this is to our knowledge the first, truly independent multi-finger online control. All right. Just get past that. And again, the performance is always as good or better if we do this with spiking band power than if we do this with threshold crossings, picking off those individual units. If you want to look at the paper, you can see all of the individual comparisons. But I can tell you, the spiking band power, it's essentially always as good. And if you have an older array, it is better. So in our lab, we've sort of switched entirely to decoding from spiking band power. And again, what that means, we worked with our circuits collaborators to simulate what that means for an ASIC, and it is a 90% power reduction. So you're going from a 30 kilosample sampling rate to a 2 kilosample sampling rate. Also, you don't have to train anything on the front end. So now you have a dumb system that can get these features. You don't have to mess around with thresholds or spike sorting or anything like that. So I think this is the point where neuroscience tools and brain-machine interface tools just part ways. We need a very different system to do this, if we want that 90% power reduction for an implantable device. Okay. For the last part of my talk here, I want to talk about electrodes. So we sort of went from, what's human and, already starting initial human experiments back through to non-human primates where, it's kind of one step away from humans. Now let's go all the way back to electrodes, which is, still happening in a small animal model, but it's something where my ulterior motive for working on carbon fiber electrodes is to ultimately impact motor neuroprosthetics. Okay, so let's talk about electrodes. What's wrong with, the conventional electrodes that we've had for 20 years? And it's the following. If I take, if I take a Utah array, where the shanks might be, 80 microns in diameter, and if I put something that sized in the brain, it's going to provoke a significant scarring response. So I have a figure here from Pat Tresco's lab where you can see there's sort of like an orange thing hiding here. That's where the electrode was. Blue dots in this top graph are neurons, and this is distance. And you can see that for about 200 microns extending away from where this electrode was, I have many fewer neurons, and purple down here is neurofilament. So something is terribly wrong with my neuron count near these electrodes. So every brain-machine interface you've ever seen is making do with what's left. It's not like it sort of starts out and then it stops working over a few months. Actually, we mostly do these experiments after 2 months. So, we are working with whatever blue dot is remaining near the electrodes. And then you have to go way out over here if you want to get to a more healthy brain. And in fact, the Utah array has shanks that are 400 microns apart. Okay, so I think a potential solution for this started emerging in the literature with John Seymour's paper in 2007. And it's, looking at what happens if you just make the thing that you put in the brain extremely small, like smaller than the neurons themselves. And so let me show you the quantification figure. So there was also a similar study from Pat Tresco's lab at Utah. So both of these involve putting a lattice structure into the brain. And they found that, if I have, only 35% of normal neuron density near this solid shank over here, I have 90% normal neuron density around the lattice shanks. And, again, these are smaller than neurons themselves. So this is really promising. It kind of suggests that, below a certain size, you can fly below the radar of the immune system. Now, of course, we don't want to do this. We don't want to put in, a million of these lattice structures, because every one you put in is doing damage on the way down, even if the electrodes ultimately end up close to the neurons. What we really want is to have something that small that you can put in as an individuated electrode. So, only something smaller than a neuron enters the brain. And I should say, in 2020, there's now a lot of approaches to doing this. I'll show a slide at the end. My group does this with carbon fiber electrodes. And so let me explain, there's a lot of cool electrodes in the world. What is cool about carbon? And I think that carbon has amazing mechanical properties, right? And this is why, you have carbon fiber tennis rackets and carbon fiber tires and things like that. And, and by the way, we're talking about the same stuff, right? These are conductive, super, super cheap. And the other thing is, they do have excellent chemical stability also. That's not so much my group's expertise, but I think they're a real contender for something that can stay in the brain for 50 years, right? And their carbon is highly biocompatible. We are carbon-based life forms. So the other cool thing, so let me just illustrate the mechanical properties here. You can actually, you can bend them 10,000 times. They do need to have some strain relief because you can't violate their radius of curvature. But if you put them in 300 microns of silicon, you can bend them 10,000 times without breaking, which I think bodes well for long-term implant in the body. All right, so our methods, started with the methods of TK Kozai, which he published in 2012, though we now actually build these in many different ways. So, but basically you have a conductive carbon fiber, you coat it with Perylene C, which we're still using, you cut the tip, which we're now doing in a variety of ways, including laser, blowtorch, and, so don't use scissors. That was the biggest takeaway from the early stuff. And then we have to coat it with something. And we're using PDOT, we're roughening up the bare carbon, and we're also using platinum with Jim Weiland's group. All right, and so again, the big benefit that you get from using carbon fiber, we published this in 2015, is, unlike all of the other, very small electrodes that have, insertion shuttles or, like the Neuralink robot, carbon just goes in, right? And it can go in at a reasonable length. If you have 500 microns of bare carbon, and by the way, these are blunt, you can just turn the screw on the micromanipulator and drive them into the brain. So what we have here is carbon, and then it can't quite go the whole millimeter by itself. So there's a little bit of silicon here that's helping it go, the last 500 microns. And it's super cool to watch this. At 150 microns like that, you can kind of watch it disappear past the surface of the brain. And this ends up with a really good histological response in long term. So I'm showing you a video after 11 weeks of implantation. My student has circled the carbon fibers, and by the way, these can be sliced inside the brain. So you can reconstruct the brain tissue with the electrodes in it because it's, it's smaller than a human hair. And green dots are neurons here, and you can see that, there's no obvious decline in neurons as you move away or as you move closer to the electrode. So let me show you that in a field view. So we're not saying there's zero scarring around the carbon fibers. You can see here on the right, or on the top, white dots are where the carbon fiber was. And then you can see the neurons and the sort of blue-green around there. But in fact, there's no statistically significant difference in the number of neurons in the top image and the bottom image. And this, this makes sense in that we are able to record nice big units. There's other things that go wrong, but if we end up with, the scarring is not that bad, and you can end up with a neuron that is very close to the electrode. I think this was 13 weeks after implantation. And we recently published a paper. So, it's not just a quantity. I know there's, so carbon fiber is not winning on channel count. We make 16 channel arrays, but I think it wins on quality per electrode because you can reliably see a spike on, the vast majority of your electrodes, which is very different than, a Utah array or, even a lot of the cool silicon probes. So my student Alyssa Welly just published this paper in Journal of Neural Engineering. She's the one that switched us from scissor cutting, which sometimes works, but sometimes gives you what you see on the left. She worked out laser cutting for the whole device and characterized the unit yield. So you can see, here's an example of acute unit yield, and then here is chronic, which we get a smaller number of animals, but we're showing you every channel. And if you have a good electrode and you stick it into the brain, you should be recording at least 100 plus peak-to-peak unit on basically every electrode. It's not that the brain is quiet, it's that your electrode caused problems. So, and I think that these cellular-sized electrodes, not just ours, really have the potential of recording neurons on every single channel and ultimately multiple neurons on every single channel. So we have, so through the Brain Initiative, we're very grateful. We were funded to develop basically a series of neuroscience tools using carbon fibers. And we now settled on 3 designs that can get you anywhere in a rat brain. And we actually use a different set of devices to do nerve stuff. But we can put this on any connector. We mostly use the Intan system since we're working with a bunch of different labs. These are still carbon fibers where, they've been used for years in neuroscience labs to do dopamine sensing. So that's something that we kind of got for free for using carbon fibers. And, if we take any of our devices and process the tips to reveal bare carbon, we've now made, a 16-channel dopamine sensor. And so we're working with Josh Burke's lab and Jill Becker's lab, Paul Garris's lab, to distribute now, 16-channel FSCV systems. We're also distributing more broadly through Yusik Yoon's Mint Hub at the University of Michigan. So this is a NeuroNex NSF-sponsored project, and it's very distribution focused. So we're trying to get devices to as many neuroscience labs as possible. If you Google us and click on carbon microthreads, this goes straight to, I have a full-time technician that's making these probes for different groups. So far, we have, this is slightly old, we've actually distributed 345 devices to date. So there's sort of a curve of like, you know, we've distributed to about, you know, 30 some labs, and a subset of those, you know, more like 5 or 6, started using them regularly. So, you know, if you're interested in using carbon fiber probes, you know, definitely contact us soon. We have, you know, a couple more years on the program, and we're trying to figure out, you know, how to distribute this after that. Neuroscience isn't a huge financial market, so we're thinking that, you know, this is going to be more of a core center than a company. But, you know, please get in touch with us if you're interested in getting carbon fiber electrodes for neuroscience experiments. So I just want to end by saying that I don't think, you know, we're not the only game in town for neuron-sized electrodes. There's now a wide range of things that are showing minimal immune response in the brain. So I'm showing work from Changxi's group, from Charles Lieber's group, and from you know, Nick Malasha's group at Stanford. And there's, you know, I think that, like, in hindsight, you know, the 2015 to 2020 era is going to be known for when we figured out how to make a wire live in harmony with the brain. And, you know, all of these things are nowhere near human use. Like, it's going to take a long time for this to get, you know, reliable and, you know, fleshed out enough to the point where we can use it in humans. But on the other hand, you kind of know it's coming. You know, we now know, like, you know, when I started in 2005, we had no idea how to make a system where you could do this and have, you know, neurons this close to electrodes 12 weeks after implant. So I think that this is going to be, you know, really promising over the next few years. And so, you know, if you're a young person getting into this right now, I think you can predict that, you know, neurotechnology is going to continue growing and become a larger and larger part of medicine as we go forward in the next few decades. All right, so I want to thank my funding sources for this. So at this point, you know, regenerative peripheral nerve interfaces, DARPA really got us. We started with DARPA hard, and now we're NIH safe. And the NSF Nielsen Foundation for our BMI work, and McKnight, NSF, and the NIH for carbon fiber electrodes. And I want to thank the people that, you know, actually did all of this work. This is our lab's right after quarantine picture. So they shut us all down and kicked us out of the lab in March. You know, so we got a lot of time, you know, writing papers, and our monkeys spent a lot of time watching Netflix. And now we're sort of back in the lab in small numbers of people. So I just want to thank everybody that I've had the privilege of working with, and my wonderful, wonderful students. So with that, I'd be happy to take any questions.
Thank you, Cindy. This has been a fantastic talk, and just seeing the whole span of going from all the human work down to this futuristic technology. There are some questions that are there in the Q&A and some in the chat, and others, please start writing your questions. We will take them. I'll start with Zach's questions. I think you answered one already, but these are both to do with control. He asks, for the first movie, this is a set of discrete pre-programmed movements you are selecting from, correct? Not continuous joint angle control. Still impressive either way.
Yeah, so for the human stuff, I started with our classifiers, and again, that's unfortunately just what we're stuck with commercial hands, and then sort of progressed into the continuous control, which is something we do. We can do it with the decahand. Like, the decahand moves fast enough for that, but really those are just science experiments in animation at this point, and it's not working as well as, you know, grasp control when they're moving around. And then the monkey stuff is all continuous. We don't do any classifiers there.
I'll follow up with that question with that. The Oser hand, is that being able to use the continuous control versus the Deca hand?
Ish. So unfortunately, the Oser hand fundamentally has modes in it. So we're working with their engineers, and I think we're, you know, hopefully creating the motivation to unlock more capabilities, but in their software, it's in one mode or another. So we can do continuous control within a mode, but there is a mode select that happens between every time you switch to a different degree of freedom.
Yeah. So following up, and again on control, Dr. Danziger asked, how many degrees of freedom are there at the BCI finger control?
So 2 degrees of freedom, and it's a position velocity based decoder with the velocity output. But yeah, it's two degrees of freedom. I know there's 7 degrees of freedom in the literature, but sometimes those 7 degrees of freedom are very correlated with each other. And this is 2 independent degrees of freedom of the fingers.
Yeah, and I'm going to follow up on that. So basically what we are calling power, so people should not get confused with power like power in voltages, but this is power in the frequency band effectively, is the
Yeah,
right?
you're right. That's really confusing. We're using spectral power, right? Like we're using that power band, and we are doing it to reduce the power of the whole system. So that's the motivation to go to spiking band power.
And it's effectively low frequency. It's LFPs.
Well, so that's the thing. It's not LFP. So Sam's paper that just came out, could also have been entitled "It's not LFP, it's the spikes," right? So LFP is coming from a population. Specifically, what we're saying is that if you filter in that band, you are actually getting a signal from a more restricted space around the electrode than a threshold crossing. Right? Because it's so heavily weighted towards the largest single units. It ends up, it's one step more spatially specific than threshold crossings, which means it's really not a population LFP in that band.
Okay. Then I have another question is to do with, from Gabriela asks, what brain structures are you targeting for recordings? Do you think these carbon fibers can be used for more subcortical structures like the thalamus?
Well, so I mean, at this point, we can get anywhere in a rat, and we're using these silicon shuttles. So for a neuroscience experiment, I think we can sort of use them anywhere. For the cortex, like sort of for the the science fiction 1,000 or 10,000 channels, we're trying to get to carbon alone. Because that is the thing we sort of think is going to be really biocompatible. It's going to last a long time period. And so we have to eventually get rid of the silicon. And I can tell you, I was showing you beautiful histology images of near the carbon fibers where there's, hundreds of microns of carbon fiber and the brain looks really healthy. If I go above that to the silicon portion, it's the same scarring terribleness that you see from a conventional probe. So we have to get that silicon out of there, and the silicon shuttle is not a long-term medical device plan. So we're doing things like sharpen the carbon fiber, we're temporarily shortening them to get them in, and I'm pretty focused on trying to get to a cortical system, with a large number of channels. But I mean, so one fact that's interesting, once you penetrate the pia, you can actually drive the carbon fiber through the whole brain, right? And if you look at Nick Malash's group, he's done a really good job quantifying this. So that pia is then holding it in place, and you can actually sort of get really far down at that point. But you need your entire array to 100% break the pia, and that's the really hard part.
So related question is, Andres Pena asks, is there any in vivo longevity data for the carbon electrodes?
Yes. So we published, so first of all, TK Kozai published, compatibility data and sort of the long-term histology. Tim Gardner did this for a bundled array. What we're doing is sort of individuated carbon fibers. I can tell you, we published histology in 2016. We were always really nervous about it because we pulled the electrodes out and you can't see any scarring, right? Like at the location. So we just had to be really, really sure we had the right brains, right? More recently, we've started, doing this with the electrodes in place where we can verify the exact location of the electrodes. And that was the data I was just showing you. That's not published yet, but, sorry, there's a new paper. There's a bunch of histology. We just do this in every paper now. There's going to be a 2020 paper, J&E, and that will have neuron counts in different rings around the electrode. So that's Paras Patel. So that should be out, if it's not out already, in J&E, and that'll show you neuron density around the electrodes.
Related question from Dr. Raj, can you remind us why no scarring with carbon, mechanical compatibility with modulus and chemical inertness is the question.
I don't think I'm qualified to say why, and I don't think it's been rigorously tested right? So certainly, you know, so first of all, even though carbon is hard, these electrodes are really soft, right? Like, I mean, they're like wisps of cobwebs as you're, you know, playing with them. So like, they're very flexible. So, you know, I think they still kind of count as soft. I'm sure that small is a major part of what's going on here, and I'm just not qualified to comment on the chemical side. You know, whether there's, you know, I've read sort of different things in the literature about whether silicon has a fundamental problem, you know, but I think others are doing a really amazing job on coding of electrodes and figuring out what sort of, you know, surface interface makes the brain happy.
Yeah, I just want to make a comment to the students listening. It is silicon versus silicone. So when Dr. Chestek's talking about, she's talking about the hard silicon part, not the silicone, which is that is often used to coat leads that are, for example, that are implanted, etc. So, okay, then two kind of related questions, and we have a few minutes left. Dr. Danziger asks, are people using your carbon fibers for peripheral nerve recording or brain only? And Satya Kumar asks, can you comment on the possibility of using RPIs for sensory feedback? So one about the nerve carbon fibers, and then moving you to a different, and you back to your RPIs.
Yeah, so for carbon fibers, yes, so we're using him in nerve, and by we I mean Tim Bruns. So he's my next door neighbor at Michigan, and Alyssa Welly, again, she's working on the paper for this. She, you know, figured out that sharpened carbon fibers, you know, below about 200 microns can penetrate epineurium. So you can just sort of drive an array of these through, you know, a vagus nerve. We've also done it at a bunch of other nerves. There's a bioRxiv paper that just that Tim just posted. First author Jiman, Jiman, that sort of just shows like the zoology of lots of stuff that we recorded using carbon fibers in the nervous system. So it's not peer reviewed yet, but if you want to see what the signals look like, you know, you can see carbon fiber signals in that. And then we're hopefully going to have another paper coming out on, you know, fabrication and characterization of those arrays. And so the second question was sensory feedback. And I would say, I think that other groups, present company included, have a, you know, what's probably a better system for sensation. We absolutely can stimulate, and we do on these RPNIs, but it's basically a nerve cuff, right? It's a cuff where the muscle is, you know, the muscle is acting where, you know, the nerve cuff would be, and we get basically the same things that you would get from stimulating on a nerve cuff. So, you know, unfortunately, it feels more like tingling. It's anatomically, you know, about as specific as you'd expect from a cuff, and we absolutely are doing those experiments. I think they're sort of enjoyable for our participants. We're doing hard and soft objects and things like that. But, you know, I have the utmost respect for, you know, the sensory groups, and I think we're very much a motor group.
So. Okay, so then, well, there could be a good marriage happening here. So, you know, so then there is some anonymous attendee who wants to know, and I guess this is your take on the, well, there's two kind of related questions. What is the progress of research in the field of visual indication of displaying our emotions, like the color of the coin changes to red when we hold it in anger state? That is one question, but an anonymous attendee wants to know, do you see neurotechnology moving beyond prosthetics? For example, do you see neurotechnology playing a role in robotics?
Neurotechnology in robotics. Okay, so let me take the first one. Emotional decoding via BMI. I was like, wow, are we there yet? Miriam Shinichi at USC is absolutely using BMI techniques to decode mood. And so the idea is eventually to develop a closed loop controller for depression or other mental illnesses. So so you should definitely check out her work. Okay, so the next one, sorry, was the question about augmentation or was the question about robotics?
No, the question is, do you see neurotechnology playing a role in robotics?
Do I see neurotechnology playing a role in robotics? Yes. I think that, you know, so I'm core faculty in the Robotics Institute at Michigan, which I found to be, you know, it's a wonderful environment. And it's a lot of people that have all of the same problems that we have in BMI in terms of, you know, your sensors, the system level experiments, all of that. right now, the only thing that we have that can control a 5-fingered soft actuator is the brain, right? In the Amazon warehouse, that's the last thing that's not being done by a robot, because that's it. It's the brain or nothing. So I think that we're obviously trying to work on these approaches to help people with spinal cord injuries and paralysis. But I do believe that, the principles that we uncover in trying to figure out how in the world the brain does this thing that we can't yet do with robots is going to have implications for robotics more broadly.
Okay. So if not directly, indirectly, but it seems like, even prosthetic hands that you are talking about, in a way, prostheses are robots and that are being driven. So, when you're talking about that, you actually are using neurotechnology for robotic control.
Absolutely. Yeah.
So.
Our definition of robots is, sensing, reasoning, and acting. And so neuroprosthetics is clearly robotics.
Robotics, yes. So, which I think not many times people think of it that way, and then are not putting it all in together. So at this time it is 10:02, and it is absolutely fascinating to see what you have been able to achieve. I personally am looking forward to see where this carbon fiber technology takes us. I know Mario has done where they're baking knots out of these carbon fibers, and they can yes, so they can do tiny knots around very tiny nerves. So there's a cuffs effectively with carbon fibers. So there's a whole another pathway that we're looking forward there. I want to tell students who are on board that you can see the excitement that Dr. Chestek has, but look at the span of being able to go from animal research all the way to human research while keeping your strength of materials, your coding and decoding information, so signal processing controls, all pulled together. And the future is really bright for the field, and we are absolutely glad. And I can also put in a word that this is the year that the women got the vote in the US, and Dr. Chestek is a woman role model for all of us in this field. So